I'm Daniel Levine, and this is the Bio Report. In cancer, the rigorous translational machinery of the human body goes haywire, causing an overproduction of proteins that fuel the growth and spread of tumors, as well as enabling them to evade the immune system. Effector Therapeutics is developing a new class of cancer therapies called selective translation regulator inhibitors, or STRIs, that can inhibit the production of proteins that drive a cancer. We spoke to Steve Worland, president and CEO of Effector, about the company's new class of therapies, how they target a central node where two major cancer signaling pathways converge, and how they pack the punch of a combination therapy in a single drug. Steve, thanks for joining us.
Oh, Danny, I'm very happy to be here. Looking forward to it.
We're going to talk about cancer, Effector Therapeutics, and its efforts to develop a new class of therapies designed to inhibit the expression of proteins that drive the growth and spread of cancer. Before we do that, though, I wanted to start with you. Prior to your current position, you were CEO of Anidis, a company developing therapies for hepatitis C, and you also headed antiviral research at Pfizer before that. Treating cancer has increasingly focused on its interactions with the immune system. I'm wondering about how you found yourself heading a cancer company and how your history as a virologist informs what you're doing today.
Yeah, well, thanks, Danny. So I think first, for the record, I should correct in behalf of everyone who's a real virologist. I'm not a virologist, although I spent many years developing antiviral drugs, but there's a whole set of skills that virologists have that I don't have. But with that said, I think the real similarity was we've had these two waves, even before COVID, we had two waves of antiviral development. First HIV in the 90s, and then hepatitis C in sort of the late 90s and 2000s. And these molecular details of the diseases are quite different, but there's one thing that we learned very important in the virals that I thought would be applicable in cancer treatment. And that was really to understand the complexity of the disease, and particularly to understand how certain diseases respond to therapy and anticipate what that response would be and then build that in. So I like to draw an analogy of you're playing chess with the disease. If I move here, that means I use drug A, the virus is going to mutate this way. If I then use drug B, this is what will happen. If I use drug A and B together, maybe I'll do better. That kind of thinking about anticipating the disease isn't static, it's very dynamic, and viruses grow very quickly and then mutate very quickly. So how will the disease respond when I treat it with therapy? And let me bake that in ahead of time in my thinking. That was what I brought over to the cancer world, and I was by no means the first person to think of this or the only person. But the idea that cancer is very complex, it's genetically way more complex than a virus disease is, and it's also very dynamic. That's one of the things we learned, is that cancer mutates all the time too, just like, not as fast as a virus, but similar thing. Cancer is not going to sit there and take the therapy lying down. It's going to fight back. It's going to change and try to escape the treatment. And so it was that thinking about how do we think about the playing field? How do we think about boxing the disease into a corner? It was that thinking in the antiviral world that when I came back to wanting to work exclusively in cancer, it was that kind of thinking that formed the basis for starting Effector.
The response to that problem, both in the antiviral world and the world of cancer, has been to use combination therapies. In the realm of cancer, how successful has that strategy been?
Yeah, I would say we've had success, and we being the community broadly, but there's still a lot of room to go. And so Most cancer therapy is used in combination. There are some exceptions for what we call targeted therapy, where sometimes there's a single gene that is so dominant in driving that cancer, all you have to do is inhibit that gene product and you'll do very well. A great example is the first targeted therapy, maybe a drug called Gleevec, targets a protein called BCR-ABL. That was so dominant in that particular disease type that's effector therapy. Unfortunately, that's the exception. Most cancer is more complex than that, and so people are doing combinations. The original combinations were a little bit sort of seat of our pants. We have an active drug, we have another active drug, maybe they'll work better together. But about the time we started Effector, people started to think about very rational combinations, which is what led to all the advances in HIV and then hepatitis C, was not just put two active drugs together, but really think hard about which combination you're doing. And so I would say we're starting to make progress. We're starting to invoke the immune system as well, but there's still a lot of complex cancers where we don't quite know the trick yet, which two drugs to combine together, or we might not have the right drug for one of the two. We like to talk about vulnerabilities in a cancer where you could hit it and cause it some pain. We don't know the right vulnerability yet for some cancers. So I think the concept is showing promise, but there's still a lot of room to go before we can really effectively treat most cancers.
Effector is developing a new class of cancer medicines called selective translation regulator inhibitors, or STRIs. What are these and how do they work?
Yeah, so these drugs are designed to block production of some key proteins that are important for cancer. So I mentioned cancer is very complex. And so for a cancer cell to grow and become a tumor and avoid your immune system, they need to make some specific proteins that would normally be in healthy cells as well, but the cancer cells make more of them and they make a particular set of proteins or a particular repertoire of proteins. So what the STRIs do is they work right at that production mechanism for protein, and they selectively go in and knock down the proteins that the cancer particularly needs. Well, because they're selective, they leave most of the proteins in a cell untouched.
How do these differ from tyrosine kinase inhibitors, and how targeted are they? Would you profile a patient's tumor to determine the appropriateness of a given STRI?
Yeah, it's a great question, Danny. And so I think compared to a tyrosine kinase inhibitor, that's kind of blocking the signals that go towards the tumor cell and say, do all the things that make you a tumor cell. Copy your DNA, make protein, get ready to divide the cell, avoid the immune system. And so while they can block that signaling broadly, there's also lots of escape mechanisms that tyrosine kinase inhibitors usually have select for mutations that after which the disease progresses. So what the STRIs do is sort of go what we call downstream. They go down to where the actual action is at the protein production plant and say, let's not make these specific proteins. Let's block the tumor from making these specific proteins. And so it's a little bit of a different approach to deny the cancer which proteins it needs in order to be a tumor.
How universal are the targets of STRIs from cancer to cancer? Are they unique to each type of cancer, or are there common targets that extend across different cancers?
Yeah, so I think in general, we think about it as there are common targets that extend across multiple cancers. Not all necessarily, but multiple. And let me give you two examples. So our two lead programs, one of them is called tummilacertib, or we call that TOMI for short. TOMI activates T cells, and so it makes T cells, and T cells are part of our immune system. They're inherently how we fight off infections and how we eliminate diseased cells. Cancer is very clever, and it sort of tunes down the immune system. It evades the immune system in part by by basically driving the immune cell into a less active state. So what TOMI can do is reverse that process, reactivate T cells, and make the T cells then go look for tumor cells and eliminate the T cells. So that's using the body's own mechanism in order to go after the cancer, which is one of the main thrusts of cancer therapy today. So to your question about would that work everywhere, it wouldn't work everywhere, but there are many different tumors that we now know are responsive to immune activation. And so lung cancer is a great example. Melanoma is another example. Kidney cancer. There are some others where it doesn't work as well. But our understanding for Tommy would be that anywhere where that tumor or that cancer is sensitive to immune activation, Tommy has a good chance of working. And then some tumors just have other ways of even an activated immune system can attack that tumor. And so Tommy might not work there. And then for the second drug, zotadifin, it works inside cancer cells. And the particular proteins we knock down are very important for particular tumor types, like ER-positive breast cancer, and again, lung cancer. So to summarize, it's not all tumors, but it would be a good set of tumors for either Tommy or Zota is the second example.
You're looking at Tommy in combination with immunotherapies. What happens when you combine Tommy with an immunotherapy?
Yeah, and so let me give a little bit of background there first. So when immunotherapy is used in lung cancer, where we're starting the study here, you see a portion of the patients respond. Some of them don't respond, and by respond means their tumor shrinks and they feel good. But over time, those tumors start to grow back again. And what's pretty clear is in most of those patients, the tumor grows back because the tumor learns a way to downregulate the immune system, to sort of shut the immune system back off again. And so when we bring in Tommy, which can reactivate the immune system, it's a very well matched with the mechanisms that the tumor is using to try to shut off the tumor system. So that's kind of the tumor's escape path. So we can shut off the escape path. And so what we've done in a clinical trial where we've published at an annual meeting called ASCO, we took patients who were actually progressing on their immunotherapy on either pembrolizumab or nivolumab. And even after they were progressing, we added our drug Tommy. And what we saw was that in many of those patients, we could stabilize the disease and stop it from continuing to grow. And we looked how long that could last. And that was stood out compared to what you might have expected from historical data. So the real idea here is to use Tommy to prevent the tumor from escaping from other immune therapy. And we're in a randomized trial right now. And so we'll have to wait till that trial reads out to know how it's working. But we're very optimistic based on the phase 2A data that we've already presented.
What do you know about it from studies that have been done to date?
Yeah, so we know that in a particular group of patients that are known to be immunoresponsive, we took a set of patients who were progressing on their immunotherapy, added Tommy, and those patients got another year on average without progression. So, that's pretty significant in a setting where people's disease are progressing, and now their next choice is really just a pretty toxic, hard to take chemotherapy regimen that won't work very long anyway. To be able to give somebody another year of their life back on average is pretty significant. So that's what we did in what we call a phase 2A study, the first study that led into our randomized phase 2B study. So we're pretty optimistic based on that data, a whole year on average extension beyond when the patients were starting to progress on their checkpoint therapy. And now, of course, we have to test that again and hope to demonstrate that in this larger randomized study.
You also mentioned zotadifin, which is another STRI that you have in development. What is zotadifin and how does it work?
Yeah, so zotadifin, and we call that one Zota for short. So our drugs we call Tommy and Zota, but they're easier to say. So Zota works inside the tumor cell, and it blocks primarily production of the proteins that allow a cell to divide. So in normal biology, cells have a cycle they go through, and they don't just wake up one morning and say, hey, let's divide and become two cells. It's a very programmed set of activities that have to follow in a sequence, and that sequence is called the cell cycle. And because when a cell divides, that's a very important event. It can be very healthy or it can be dangerous like cancer. It's a very tightly regulated process. And so there are proteins that control advancing through this cell cycle to ultimately divide the cell. And so what Zota does is it blocks production of those proteins that are required to advance through the cell cycle. And what we've learned is that that's a very important aspect of cancer. It gets hypercharged in cancer, and particularly cancers like a set of breast cancers we call ER positive, meaning estrogen-driven breast cancers are very dependent on this cell cycle. And people have previously shown other ways of going after the cell cycle work in breast cancer. So that's one of the main focuses for Zotadefin now, is to use these properties of blocking cell cycle protein production to treat breast cancer.
This is in development for solid tumors. Have you narrowed down the indications you're pursuing at all? And how are you going about determining those indications?
Yeah, so that's a really good question. And, I should point out that we're really the leaders in the STRI field. So every drug we're taking into the clinic, we're plowing new ground ourselves. So no one's ever taken a mechanism like Zota into the clinic before. So what you usually do is you have some idea from your preclinical studies, and you enroll a fairly broad set of patients, and then you start asking, what's happening in the clinic. And so what we've seen is we've seen 2 responses so far. We reported this at the ASCO meeting in June. We've seen 2 partial responses, meaning tumors have shrunk beyond an objective threshold in these patients with estrogen-dependent breast cancer. So the way you figure this out is to kind of take your preclinical data, use that to shape where you start in the clinic. And that's kind of a bootstrapping thing. The more you learn in the clinic, the more you hone in on particular tumor types. And so right now we're very focused on ER-positive breast cancer, and are very excited about the first 2 responses we saw. And I'm particularly excited because it matches very well with the preclinical biology. And that's an important confidence builder. If what you understood about the drug before you went in the clinic matches what you're seeing in the clinic, that's usually a suggestion that clinical data is much more likely to hold up over time. If it's completely different than you saw preclinically, that might be interesting, but you probably have more work to do. So we're very excited that the clinical data, as it's starting to emerge, fits very well in our picture of how this drug, we might expect it to work, and fits very well in the picture of the overall STRI platform. So again, it's the first company to take a class of the Zota mechanism into the clinic. I think that's very exciting and very rewarding to actually see patients benefiting even this early on in the program.
What's the development path forward?
So I think we'll continue to do some of this, early exploratory phase 2 work here. But as data continues to come in, we hope that it will confirm this initial signal of activity in ER-positive breast cancer. And then we would move to what's called a randomized trial, where we would compare our drug to a different treatment and then really demonstrate in a very rigorous randomized way that we're providing more benefit than the other, what we call a control arm. And that with that data in hand, then we'd go to the FDA and propose a registration trial, meaning we want to run one more trial and get this drug approved to go on the market.
Effector went public through a SPAC deal in 2021. Why did you decide to go that route rather than a conventional IPO?
Yeah, it's a great question. And when we first started looking at it, I asked myself, why does this other avenue, this other product exist in the market? As I educated myself and had people help me and teach me, what became clear was that if you go down the SPAC route, at some point you have what we call in the business world deal certainty. You can say at a point in time, 5 or 6 months before the SPAC closes, there's a very high likelihood that this transaction is going to close. And so you get through all the risk early on. In an IPO process, I've done an IPO previously as well, about 15 years ago or so. You don't really know until 2 or 3 weeks before you have a pretty good sense, and you don't know for sure until it actually, you price it the day of the IPO. So in about 18 months ago or so or now, it looked to us at the time that there was much better deal certainty. We'll know that we'll be public. We'll know that we'll get the capital that we need to continue to develop our products. And yes, perhaps we can do an IPO. I think we could have, but there was higher risk around the chance of closing an IPO relative to doing the SPAC. And so that's really the differentiating factor for us was this deal certainty.
How far will existing cash take you, and are there plans to raise additional capital?
Yeah, so the existing cash that we have will take us through the trials that we're talking about. So we have this randomized trial with camonsertib and pembrolizumab, and that data we expect to read out in the first half of next year. And we have the ZOTA trials that I talked about, particularly focusing on ER-positive breast cancer. And so the cash that we have on hand now is enough to run those trials and get that data. We haven't commented, and we usually don't specifically about plans to raise capital, but like all biotech companies, it's a very capital intensive business. So at some point, certainly we'll want to raise more capital to finish developing the products. But the cash we have on hand right now is sufficient to read out the data events from the trials we're doing.
It's been a tough time for public biotechs. Effector is near a 52-week low. What's the discussion been like with investors, and how has being a public company made your job tougher?
Yeah, and so I think you're right that the market right now is a very risk averse, and so there's kind of a risk on appetite, risk off. We're very much in a risk off appetite right now. And I think, our investors and other investors that we talk about kind of understand that. And, people who've been in the business a long time know these cycles come and go. I've lived through several of these cycles before, and they do eventually go away. So what you have to have is enough capital to get to your data events, and investors respect that. So I think everybody is kind of feeling the pain of the current market for sure, a little bit of commiseration, I think, but also focus on companies like Effector who are going to be able to read out their clinical data events. And then, in terms of tougher or not, they, being private and public, they each have their challenges. I think for the stage of our company, I'm actually glad we're public, even though this is a very tough market to be in, and wouldn't wish on anybody to enter the public markets right before the kind of downturn we've had this year. But I still think for sort of clinical stage of development in biotech and pharmaceuticals, the investors who typically participate in the public markets are very knowledgeable about drug development. They're very knowledgeable about what kind of trials will read out useful data, and really think kind of about new products and how they'd ultimately go onto the market and be used. And so I do think it's appropriate at our stage of our company to be speaking with that type of investors. And, this, like everything else, shall pass. At some point, we'll be in better market times, and everyone who's been through these cycles knows that is the case.
Steve Worland, President and CEO of Effector Therapeutics. Steve, thanks so much for your time today.
Danny, thank you very much. I really appreciate the time.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.